Good evening, everybody, or afternoon, depending on where you're calling in from from our country. I'm Kristen Angel. As Sandra mentioned, I'm the Associate Director of Advocacy at the National Organization for Rare Disorders. I oversee our Rare Action Network program, which is our grassroots-based advocacy network that serves to connect and empower individuals with tools, trainings, and resources to be effective advocates for rare diseases through national and state-based initiatives across the US. We work on equitable access to timely diagnosis, treatment, and care for every person impacted by rare diseases. Just a quick housekeeping note, if you have questions throughout tonight's presentation, if you can please submit them through the chat box located at the bottom of your screen. There's a chat icon. And we will be addressing questions at the end with whatever time we have left after the presentations. So I'm going to get started. Patients and families living with rare disease face a great challenge and a lot of commonalities and struggles seeking diagnosis, therapies, treatments, even specialists in their rare disease. And in this session of advocating for answers, we aim to build awareness about the specific issues and challenges, as well as offer guidance and direction towards solutions and improved outcomes. So I'm going to talk a little bit about rare diseases as a whole. NORD is an umbrella organization. We represent the entire rare disease community with the work we do. And to go to the very basic, a rare disease is defined as a disease, syndrome, disorder that affects less than 200,000 of the US population. It's slightly different in the European countries, but it is approximately 7,000 rare diseases, and approximately 25 to 30 million Americans are currently living with the rare disease, which is almost 1 in 10. Most rare diseases are genetic or have a genetic component. There's more than 90% of rare diseases are without an FDA approved treatment. There are many common challenges amongst the rare disease community, and this is where NORD helps bridge a gap with the average diagnosis of 5 to 7 years for an accurate diagnosis. There's few medical experts and not easily accessible medical experts. Little research or known about these diseases. There's small scattered patient populations, so they're small numbers, and patients are scattered throughout the world, and there's extensive and lifelong medical needs. There's high cost of care and treatment, and one of a big issue that we've seen, especially recently with the pandemic, has gotten even more attention, is social isolation. It's very common for rare disease community members to feel isolated and alone because they have not necessarily been able to connect with individuals that have gone through something similar. NORD advocates on behalf of these people. These are some of the things we work on at our organization, from advocacy to patient assistance programs and support to the rare disease community. I'm going to take some time now and introduce our panelists for this evening. I'm really excited to have everyone here. I'm going to introduce them and then give them a moment to introduce themselves and tell a little bit more about themselves. So we have an incredible list, and we have Andrew Rankin is the Vice President of Global Clinical Development. Andrew, would you like to take a moment and introduce yourself?
Yes, good evening, everybody. Thanks for the introduction. So I'm involved personally with a child with Cat6A, and also have been developing medicines for rare diseases for the last 25 years, and been fortunate enough to be involved in the regulatory approval of 3 therapies for rare diseases. It's a pleasure to be on the panel this evening. Thank you.
Thank you. Now I'd like to introduce the chairperson of Cat6A and a parent, Jordan Muller.
Yeah, thanks, Kristen. Hi, everyone. I'm Jordan Muller. As she mentioned, I'm the chairman of the board of trustees for the Cat6A Foundation, and parent of 13-year-old daughter, Chloe, who was, to all our understanding, the first patient diagnosed with KAT6A positively through a genetic sequencing result. So, and that was back in 2013. So was involved in some of the development of the initial patient community from that point forward, and intimately with Emil and Natasha as they created the KAT6A Foundation. Thanks.
Thank you. Now I'd like to introduce the co-founder and parents of KAT6A Foundation, Natasha Esper.
Hi, everyone. Jordan was talking about history. So 2013 was Chloe's diagnosis, Peter 2016, and we met Jordan October 21, 2016, actually, because it was Peter's birthday. My son will turn 13 soon, and we met Jordan on that day for the race, actually, for rare disease at Children's National. So I'm a doctor, I'm an internist. That's my regular job, internal medicine physician. And since my son's diagnosis, I have been involved a lot in rare diseases with NORD. I was a speaker twice for the NORD meetings. I wrote the summary for NORD on the website for KAT6A, had one peer review article published. It was mainly from an international group who published in 2020. And I helped through the foundation. So my son is almost 13, and my goal is to find a treatment for him as soon as possible.
Thank you. I'd like to introduce Dr. Alejandro Iglesias of Columbia Presbyterian Clinical Genetics.
So now non mute. So thank you, Kristen, for the introduction, and thank you the other panels to be here tonight. So, well, as they said, I'm a clinical geneticist, also biochemical geneticist. I've been doing this for at least 30 years. And well, I can give, some of my experience in terms of the challenges and the opportunities that I think was Kristen was mentioning before. And I think that the second part is probably the most important one, because, from the challenges, we find opportunities and we move forward to help these children. Thanks.
Thank you. Now I'd like to introduce Dr. Jacqueline Harris, the Assistant Professor of Neurology, Pediatrics, and Genetics at Kennedy Krieger Institute, Johns Hopkins Medical Institutions.
Hi, everyone. Happy to be here. Thanks for having me. I am a physician, a child neurologist by training, and a researcher. And KAT6A is one of my focus areas, both clinically and research. So thank you for having me.
Thank you. And we will be actually showing a video a little later on during the presentation of Dr. Yehuda Asaroff, head of the Fred Wyskowski Cancer Research Laboratory at Technion, Israel Institute of Technology. We'll be sharing a video interview that we did with them. So I'm going to get started by introducing our first, topic, building a community of influence, the founding of the KAT6A Foundation, a rare disorder. So Natasha, I'm going to pass the mic over to you. You are the co-founder of KAT6A. Can you talk to us about your goals and journey, both as a parent and as a co-founder of the organization?
So, at the beginning, to start the organization and foundation, it was hard to find other family willing to volunteer and put their time from, away from the families, away from the special need, the child who needs extra help, and not just their time, their money. This was the main challenge at the beginning. And, we had to use a lot of our time, and we were lucky to find 5-6 other families dedicated to start it. But this was the main challenge, is finding people who already have their own families, their own jobs, their own challenges with a child with multiple health issues and behavioral issues, to come and start a foundation from zero, from scratch. And pay money, not be paid, because even till now we're still, we're still not paid. We do it all for free, and we still put our time and our money in the foundation. So this was the main, the main struggle starting.
So can you tell us why you decided to start the organization?
Because of my son's diagnosis. My son was getting worse and worse. He was kicked out of school. No school would take him. Even at home, it was hard to manage his health issues. We were, in the emergency room every other day, or in a hospital, or visiting a specialist. So there was no option other than looking for answers, meeting other families, looking for therapies, so he can go back to school and can stay in the community and the society. And fast forward, this was 2016, we're 2021 now. He is in school and he is still at home, and he's now home with his 3 siblings and the babysitter. So I guess we found something, and we were able to treat and meet researchers and meet other families, which was very, very important. Connecting with other families who have children and adults with the same syndrome. This was very helpful.
Great. I working at NORD and working through our organizational outreach that we do, and working with the nonprofit organizations and I know it is a lot of work to build an organization. So, building kind of an ecosystem of support for Cat6A and B families, I'm sure wasn't easy. But what strategies and tactics and tools did you find most successful in building awareness and gaining support to build the organization?
It was, hard to recruit at the beginning researchers and doctors from everywhere. That's, again, it's a rare disease. My son was 39 diagnosed. So Jordan's daughter was number 1, 2013, 2016, we were 39. How many researchers will be interested? How many drug company will be interested in talking to us? Or even doctors who know, since, we were only at number 39. So this was a big struggle, but social media, I know social media has pros and cons, but social media helped us a lot connect with families from all over the world. And then families connected us to their doctors, their researchers, from mouth to ear, but on social media, connecting, and then now we have research in Spain that we found. We have research in Australia. That's all because of families. Without families, I cannot just pick the phone and call a researcher in Spain. It was the families in Spain who connected the foundation with the doctors there. Same with Australia, same with Canada. So social media and connection with families helped us connect with doctors and researchers all over the world.
That's incredible. So I know you mentioned Jordan, and I'm gonna shift over to Jordan Muller. Jordan, you were the first diagnosed case with your daughter Chloe. And as the chair of the board, you've actually had some breakthrough moments. How did strength in numbers begin to affect funding and build momentum for research for the organization and for the disease?
Yeah, thanks, Kristen. So thankfully, as our patient community has grown, it's also enabled us to reach, as Natasha highlighted, reach a broader community of families, and to gain more and raise more funding over time. Stepping back to 2013, when my daughter was first diagnosed with exome sequencing, I want to highlight, it's fortuitous. We live in the Washington, D.C. area, and I'd say only because of that geography did we become that first family. There's clearly patients much older than Chloe, in their 30s and 40s even, But she was first diagnosed because our geneticist at Children's, a world leading children's hospital, Children's National Medical Center here in DC, our geneticist, who is Ken Rosenbaum, super experienced, seen tens of thousands of children. He was very well connected with the genetics community around Washington, DC, which was also fundamental in the first sequencing of the human genome, over decades ago. So he was able to see Chloe, understand while she didn't initially have a diagnosis, when she was 5 years old, he reached out to his contacts at the local genetics labs here and was able to get her sequenced back in April 2013. So when they found that CAT6A mutation, there were obviously no other patients known to have a global developmental delay associated with that mutation. So we had to wait 9 months. And when he called us back one night and said, "Oh my gosh, I've just learned of 3 other children elsewhere in the world that have the same mutation and similar global developmental delay." So from that point forward, created a website with my contact information, search in Google CAT6A, you hit it, families started contacting me, like Natasha and Emil. And quickly I was able to start raising some funds through the existing fundraising platforms at Children's National Medical Center. But quickly, as Natasha highlighted, they came down to the race, but that was fundraising for the Children's Hospital in general. Even though it was earmarked for CAT6A, it was very difficult to take the funds we were raising through that organization and apply them and send them to the one researcher at the time who had an interest in CAT6A. So it really highlighted when we were a small group, tens of patients, leveraging the fundraising of an organization not specifically focused on CAT6A. It was really challenging. And so I think it highlighted the need to create a foundation, a family-run foundation, in order to raise funds and directly administer those funds to researchers that we felt were on point with where we needed to go, and to provide some funding directly for family support. And that grassroots fundraising we were able to do once we started the CAT6A Foundation, that's really served as the platform. And sorry, Kristen, I might be bleeding over in your second question here.
That works. No, you can continue, because that was going to be kind of my follow-up in terms of grassroots. I know that word's been used a lot so far tonight, and even in the work I do is grassroots. So I mean, grassroots is really feet on the ground, pounding the pavement, knocking on doors, knocking down doors. So can you talk a little more about the efforts in your traditional channels for fundraising? Like you mentioned a walk, but also through grants, conferences, and global connections.
Yeah. So again, we first started doing this run through Children's National Medical Center, and then as soon as Emil and Natasha founded the CAT6A Foundation, some of the families started organizing a catwalk, which again is like a grassroots annual walk event where it's largely friends and family consistently donating year over year, but it's still a relatively small effort. But importantly, it did provide a base of operations, the funds we raised through there, to support some foundational things that the foundation does, like support our patient registry, for example, and the maintenance and recruitment and marketing of that. But then most importantly, that grassroots fundraising then allowed us to apply effectively for a 2-year grant opportunity through the Chan Zuckerberg Initiative. And we were successful in that, one of about 30 rare disease organizations that successfully received a 2-year award expressly to build out our research network of researchers and clinicians interested in CAT6A. So I think we've been kind of a model example of starting before a formal foundation, raising some initial funds, creating our own foundation, and then using the grassroots fundraising through the new foundation to then start applying for grants and external funding. So we've got a good track record so far, but clearly I think we're ready to try to take that next step to get more formal, regularized, ongoing annual funding beyond, the grassroots level.
Great. And Natasha, do you have anything to add before I move on to the next speaker?
No, we're constantly applying for new grants. We're not stopping. I think what pushes us is our children. We're not just doctors or researchers or just we want to find answers soon. So we're pushing. We're always applying for new grants.
Great. I think for some of the folks on the call, just in my experience, some of the things that you can do to help the organization push through is helping bring that awareness. I mean, awareness is part of this process and building an organization and getting people supporting it. So, just by sharing your stories and going out into the community and talking about it and, letting people know this organization is out there is a really big part of that process. So, that's just kind of something that you all on the call can take home and realize how you can contribute as well, and even just a small way by sharing your story. Great. So we are going to do, we have a short video interview, sorry. Karen Ginsberg is interviewing Dr. Yehida Asaraf, speaking points on his research and the potential for treatments, as well as how his research connects to other rare disorders and even common diseases.
Hi, my name is Karen Ginsberg. I have a 13-year-old daughter who was diagnosed with the mutation on her KAT6A gene in 2015, and we've been on a remarkable journey ever since then. And I'm thrilled today to be speaking to Dr. Yehida Asaraf at the Technion Institute in Haifa, Israel. He has been doing some very groundbreaking work on KAT6A and is an expert on the gene. So let me first start by asking you, Dr. Asaraf, just to introduce yourself and how you came to be part of this research.
Basically, I think I would say that in the past 4 decades, we have been studying the molecular basis of human diseases, including cancer and genetic diseases. In the past, basically 3-4 decades, we've been focusing a lot of studies not only in human genetic diseases, but also on cancer drug resistance. Along this very vein that we do this the deciphering of these genetic diseases by pinpointing the very complex mechanisms of disease, okay? And the syndromes and the symptoms that we see in individual patients, we believe that we can tailor-make therapy and interventions. So we are very optimistic towards the so-called precision medicine, personalized medicine in the individual patients.
Yehida, can you give us a sense of the research you've been doing, the KAT6A research you've been doing for the past 5 years?
In 2016, I actually was introduced to Sam and John Ginsberg. Sam and John Ginsberg are Karen's parents. Sam and John said, well, we have a granddaughter by the name of Lexi, which is harboring KAT6A syndrome. And I said, wow, KAT6A is a lysine acetyltransferase 6A, which is a maestro regulator of gene expression. We are very knowledgeable in this field. And I knew I could contribute to it because I'm knowledgeable in the studies of molecular basis of gene regulation, etc. So we started epigenetics, genomics, i.e. DNA level, then moved to transcriptomics. You remember gene, then mRNA level, we call them transcriptomics. When I said transcriptomics, I mean any messenger RNA level in terms of quantity and quality. What genes are we talking about? So this actually is pinpointing what genes are altered and to what extent. So qualitatively and quantitatively and vice versa. Now, we also inserted a very, very unique approach here. Beyond the proteomics, we asked ourselves, since KAT6A is such a maestro or master regulator, we want to know who are the client proteins that it actually identifies. So we're talking about KAT6A interactomics. Interactome means who do I interact with? I'm Mr. KAT6A. I want to know what clients do I have. That's the way we can actually pinpoint what client proteins it actually addresses in a manner that would tell us what pathways could be affected and what alterations are we may be seeing and what potential remedies could we bring in, because those are deficiencies that would be in place. So by pinpointing the certain protein interactions of KAT6A, we could also find the pathways that may be altered.
Can you let us know what kind of KAT6A research outside of what you've been doing might benefit the work you're doing? What do you think might be complementary to what you've been studying?
Pinpointing the genome-wide SNP alterations that in multiple KAT6A patients would facilitate the discovery of the genetic determinants which may be exacerbating the disease symptoms. Because the moment a kid is diagnosed with KAT6A, all is done by the geneticist. Is there a mutation in KAT6A? What is not done is, on top of KAT6A mutation, what other alterations as SNPs, alterations, okay, or phenotypic alteration, I mean, genetic alterations or variations are present which have pathological impact, especially when combined with KAT6A mutation. This is a major project which I think would bring a lot of information and benefit to the KAT6A community.
I heard you say also that that one lab can't sort of generate all the kinds of cells that might benefit from a differentiated iPSC cells. Would it also be beneficial if other labs were, coordinating with which cells they're going to differentiate?
There's no question in my mind, after 4 years of biomedical research, that coordination is important. To some extent, at least knowing which lab is doing what, and perhaps trying to share some cells, if possible, or iPSCs, that would enormously help, because you can imagine if I had now in my hands, as we discussed recently, more iPSCs from different patients, that would make a huge difference, okay? We are fighting daily on each patient, recruiting a patient and having cells from a patient is not so easy.
So on the flip side, how do you see the work that you have been doing benefiting the larger research community, whether it's the KAT6A research community or just the larger genetic research community?
It would certainly benefit people studying various, this is just an example, autism spectrum diseases, and as well as infant genetic diseases with similar and maybe less similar syndromes, because it's a very, very systematic approach that reveals a lot of information. You can imagine that we, the scientists doing all this mechanistic study, is like coming to a car mechanic and he will diagnose the problem. Of course, will charge us, okay, but then fix the problem. Once you understand how this motor is working, you can fix the motor and any problem with it. That's exactly what we're doing. But the only thing in biology is the motors are the most complex that you can even think of.
And now I want to ask you a bit about treatments and how you see, what directions you see the treatment possibilities in. And I'm wondering if you could maybe speak to nutraceutical benefits or dietary benefits, and what possible treatment avenues you see emerging.
I think the plethora of findings that we had, that there is some clear-cut bioenergetic deficiency metabolite alterations, oxidant stress, oxidant markers, antioxidant deficiency, and more. I'm a very avid believer that nutraceutical interventions prove already and may further prove highly beneficial and effective for CAT6A syndrome. And I would say more than that, that the deeper we're going now into the molecular pathways and mechanisms that are underlying this disease, okay, the more we see that, as I said, it's just put it in a most layman manner, that it's like a mechanic looking at a complex motor, and the moment you are starting to understand better and better how this motor is working, functioning, you don't need to see the entire picture to be able to help. The example I can give you, the first antibiotics came 90 years ago, okay, almost 90 years ago. There was very little knowledge about the mechanism of action of these antibiotics, but we cured patients. There was very little knowledge how the antibiotic works. They put it on a bacteria, it died. Put it on a normal cell, human cell, nothing happened. Oh, selected, great, let's treat. And it worked. Many times, scientists can actually address and find remedy much before they have the entire picture. You always have to say, how much of the picture do I have to give me the tools to be able to intervene in a proper manner? When you deal with such a syndrome, it is so complex, it takes more than a single soldier, obviously. We need many soldiers on our side and as many groups as possible to actually cross-feed one the other, and it helps a lot. I think we should be aware that these studies are extremely costly, and we need always to be able to try to fund them, okay?
Okay, well, thank you. So I'm going to introduce Dr. Jacqueline Harris, and I have a specific question. So Dr. Harris, as a researcher who's recently participated in the Global Research Symposium for CAT6A and CAT6B, you also published a paper recently. Can you talk to us about what made you go into research for CAT6, A and B, and what kind of, how you got there?
Sure. So as I mentioned, I am a neurologist by training, and but all of my clinic work and research work is in epigenetic disorders, of which CAT6A is one of them. And I am extremely interested in the fact that all of these epigenetic disorders, like CAT6A, or what we call, more specifically, what we call Mendelian disorders of the epigenetic machinery. So CAT6A, the protein, is an epigenetic machinery protein, that they all lead to significant cognitive dysfunction. And that may be speech most affected, like in CAT6A, or it may be other patterns in these others, but they all lead to significant cognitive dysfunction, which means that they have a strong role in the brain. I've also been fascinated by the fact that the basic science work suggests that these epigenetic disorders, like CAT6A, may actually be treatable in postnatal life. And what I mean by that is that some of the cognitive dysfunction or brain dysfunction may be reversible even after the child with the disorder is born. And that is contrary to what we used to believe is possible, but now believe that it is possible. And my main area of interest is something called translational research, which is taking what's found in animal models and cellular models and translating that into studies in humans to prepare for and then eventually carry out clinical trials looking at some of these treatments that we think may be possible. So other researchers you may hear from focus a lot on the specific type of treatment, and that's very necessary and wonderful. And I focus a lot on, okay, once we get whatever treatment X, Y, and Z, because as Dr. Asaraf just said, it may be necessary that we may need to treat different pieces of the disorder with different things, but whatever treatment it is, that we're ready with outcome measures and biomarkers and design of a clinical trial to carry out a clinical trial to test whether these things really work. And so that's where my area of interest lies. So KAT6A fits in very, very well with that area of interest. And then in addition, early on in my career, I met Natasha and Emil, and then eventually a lot of other KAT6A families, and they were very motivated and a wonderful group of families and patients to work with. And so I became very involved in the community. I started directing the conferences that have, other than last year because of COVID, have taken place every year for several years in Baltimore, where I work, and have just been very motivated to stay involved with this disease and this group.
That's great. So relative to your research that you've done and what was uncovered at the Global Symposium, what are some of the highlights?
So I think that a lot of what was uncovered at the symposium was that there are a lot of people working in different areas within this, at least with relevance to this disorder. And that's very exciting. And so we need, as others have said, sort of a roadmap of research that's going on in the disorder to foster and enable collaboration. And we need people collaborating so that it's not, of course, all research needs to be validated. It's best if different groups can come up with the same findings to know that research is true. But we don't want groups necessarily competing to find the same thing over and over again. We want people working on different things in different areas. So as I said, I'm a clinical and translational researcher, but my work is irrelevant, right? If somebody else doesn't find specific treatments, doing more of the basic science research for me to test within the clinical trials, and vice versa. You can find all the treatments in the world, but if you don't have well-designed, ready to go and very good and specific to KAT6A studies for clinical trials, then those treatments don't do much good. So so that's kind of what was highlighted. Is it's very important, the research that everyone is doing, and it's exciting, but it's also important to do things like this to create more formal networks so that everyone has a roadmap of the landscape and can do more formal collaborations with each other.
Great, thank you. And I apologize for the interruption. It's a sad point of COVID and working from home. So the symposium was a 90-minute webcast of research findings from around the globe. Can you explain the different perspectives and the results? And if you were to summarize the research findings from the symposium, what would they be?
Is that still for me?
Yes.
Okay. Yeah, I think that, like I said, that there are, to summarize the research findings, there are several exciting things going on as far as looking at the genetics of KAT6A and the epigenetics of KAT6A from a basic science perspective. So looking at the somewhat of what Dr. Asaraf mentioned, looking at other genes that are affected by KAT6A's function, so downstream effects, and also looking at kind of what we call the proteomics of KAT6A, or how that protein works in complex with other complexes to carry out its function, because a lot of those other genes and other proteins may be the target of potential therapies. It's clear that there are a lot of potential therapies from things like skipping part of the KAT6A gene that's dysfunctional to see if the protein could still be made, to looking at downstream metabolic effects, to looking at epigenetic modifiers, so sort of correcting the problem that the KAT6A causes from another angle. All of that is happening. And then we presented some data on kind of the functional profile of KAT6A, because before you can treat a disorder, it's really important to know about what it looks like. And so we presented some findings on adaptive behavior and sleep and maladaptive behaviors in KAT6A. Another group presented some really interesting findings on speech disorders and the specifics of that in KAT6A. And another group presented sort of a general cognitive profile of KAT6B, which hadn't really been published before. So there was a lot of different things presented, and it was very, very exciting to see the breadth of the work. But in order to really go into depth, it's important to, like I said, make sure that to formalize those collaborative networks, and also to, just like everyone said, none of this work, not mine, not anybody's, can be done without the funding to carry it out, unfortunately. So that is extremely important.
Absolutely, and that's done through the grassroots network and driven by patients and families as well, getting that support. Thank you very much, Dr. Harris. So I'm going to switch over to some questions with Andrew Rankin of Global Clinical Research. Can you talk to us, based on your experience on the corporate side of clinical research and drug development, what specific advice would you give to individuals in terms of uncovering support and innovative pathways relative to treatment methods?
Thank you, Kristen. Well, actually building on what we've just heard from Yehuda and Jacqueline, and by the way, I've had recently a number of really great conversations with Yehuda on how we can work together and join him with other researchers interested. Whilst I'm, primarily involved in the clinical development of new medicines for rare diseases, it's clear from what you've just heard that the first steps, really, the building blocks are, and it's true for rare diseases and common indications in larger populations, is understanding the basic biology underlying the condition, including the genetics. So in practical terms, identifying biotechs and startups that have an interest in diseases that, if not KAT6A, are similar, maybe mitochondrial diseases, for example, would be a good start. I mean, opening up discussions with the leadership of those organizations. I can tell you in my professional life, I work very closely with NORD, with patient foundations around the world, and understanding the disease, learning from the patient populations, the people living with rare diseases is extremely important to making progress in developing new therapies. Similarly, with academic groups with common interests, and how we can partner with them, be it through funding, sharing that kind of knowledge that I've spoken about, providing samples, which are not always easy for companies to get hold of, such as fibroblasts in the case of KAT6A. There are a number of ways, but reaching out, networking, and starting to talk to companies, startups, biotechs that have those common interests, and they will really value understanding the disease and the impact of the condition to be able to move forward.
And kind of going off of that with the techs and the startups, I apologize, late in the day for these guys. How has the corporate landscape changed to address the growing number of rare diseases and the increase in startups, biotech firms, and how the industry sector is evolving in this way?
Yeah, that's a great question. There are companies that now specialize in developing new therapies for rare diseases. Actually, many companies like BioMarin and Ultragenyx on the West Coast of the United States. And most big pharma companies have also set up their own rare disease business units, such as Pfizer, for example, often in the press these days, or acquired rare disease companies. AstraZeneca recently bought a company that I worked with previously called Alexion, which was a rare disease company. So they are very aware that it's specialist knowledge and a very different way of developing medicines. The regulatory path is very different. So companies have both grown to meet that need, and also the larger companies have acquired what they need or develop their own business news. Now, this is a bit of a double-edged sword, because while it's very good news for the rare disease community, the increasing number of drug development programs, new medicines for rare orphan diseases being approved, and these have very high price tags in general. Why? Because it costs just as much to develop those drugs, but there are very few patients who can access them and be reimbursed. So the price becomes very high, and this is putting increasing pressure on payers and healthcare systems, particularly ex-US, where there's social medical systems, but also even in the US. And these payers and healthcare systems are having to make very tough decisions, healthcare economic decisions. So industry is looking more and more carefully on which orphan rare diseases to prioritize. Looking at the scientific knowledge, identifying drug targets, whether they can actually identify a drug target, the size of the patient population, and whether it's feasible to run a clinical trial. What is the regulatory pathway to approval of a medicine? And beyond the regulatory pathway, because of the high price tag, a value proposition. Really, you need to be able to differentiate, show that you're having real both economic and quality of life impact on the rare disease patients. But this is what companies are dealing with, and I think how the industry is evolving.
Thank you so much. Appreciate it. I'm going to introduce Dr. Alejandro Iglesias. You are a pediatric geneticist who's been responsible for diagnosis and treatment of patients with X-linked adrenal leukodystrophy. Could you talk to us about newborn screening? And interestingly enough, NORD has been promoting all month that September is Newborn Screening Awareness Month. So it's a great time to kind of talk to us a little more about newborn screening and the work in New York that's made a difference in developing more pathways to cure and research funding.
Yeah, so thank you, Kristen. So I think, the adrenal leukodystrophy story is quite interesting in terms of newborn screening. And I would say in general, as a simple statement to introduce this. So the whole purpose of newborn screening that, started, at the beginning of the 60s with, PKU, it was just to, after identifying a disease and identifying a treatment for the disease, to try to figure out a way to detect the disease before the symptoms start. And this is, in some venue, what Dr. Harris was saying, that although, we might treat patients after they're born and after they actually have symptoms, well, I would say if we can treat them before they develop symptoms, it's, theoretically much better. So the PKU story is, a massive success, not just in the US, worldwide. So since we started and we detect this, We have, adults in their, 50s, 60s, healthy people with their families, their children. So from there, the thing evolved over time. And of course, the same rationale was used for other disorders. And I would say until probably the 70s, it was kind of a little difficult to test for things. So we were using very short panels. It was like 5 or 6 disorders in the US. But eventually in the 90s, technological, developments allowed to use a single machine to test for like, initially 35 or 40 things related with each other, but eventually the machine was kind of tweaked. And so now we can use the same machine to test for, at least in New York, for probably close to 65 things now. So somewhere along this, the experience with, adrenoleukodystrophy, and Dr. Harris, doing this very well because she's at Hopkins. So with, the massive work that, Dr. Moser did down there and, all the colleagues working with him to figure out there was a way to treat this disease. I guess ALD has, this unique feature that even if you have the disease, it takes few years to potentially develop the disease. And so, and they have a large population. And this goes back to, I would say, everything that everybody has been saying here. So we need, a village to take care of our kids. So all the players here, and of course, the parents advocating and, creating these foundations, especially for the things that are super rare. And as you were saying at some point, well, usually people don't want to put money there. So if you look for grant support for something super rare, well, probably you're at the bottom of the list. It's other people before you. So eventually we need to look somewhere else. So the parents are, a source of this, creating these groups, foundation, linking everybody together. So as, Natasha was saying, social media is good and bad, but I would say in this case is wonderful and helps. But the industry, as he was just saying also, it's a big player here, especially for super rare things. So the experience there was able to detect kids at birth because they have siblings that were already diagnosed. And they thought, well, maybe if we treat these kids before they develop symptoms, they won't. And that was actually true. The treatment was, initially, bone marrow transplant. It works very well. The disease gives time to find donors and all this. So the logic, conclusion of this was, okay, if we can eventually detect these kids at birth or soon after birth, we can start just identifying kids that don't have family history because, the family history was for small groups. So what about the rest of the population? Well, eventually, again, for, Dr. Moser's wife and the group from Hopkins was so advocating for this and all the parents. And eventually, the state agreed that this was a good idea. So we started this back in 2014. So interesting enough, I got the first patient in New York State, like, January 2nd or 3rd, something like that. And this family has, a complete healthy kid, wonderful. And this is why probably the families play a huge role. The mother of this boy is actually a huge advocate for this. And she runs, one of the foundations. She's available for other mothers and other fathers to talk to her. So one thing led to the next. And the experience that we have, and other states, of course, eventually follow, has been, wonderful. So this has been, a tremendous success for a disease that in boys was, I would say, probably 50% of them lethal because, when they started with the symptoms, it was no way to cure them. I mean, Lorenzo's oil was used and still some people use it and delay the disease, but it doesn't cure the disease. So meaning that, from that point of view, this is a really powerful story of a success of a newborn screening. Yeah.
Thank you, Dr. Iglesias. I can speak a little myself as to, the newborn screening process, and it is definitely something that NORD has taken a priority in our advocacy work, both at the federal and state level. And it is a process to get a disease listed to be screened, on the Department of Health's RUS recommended screening panel.
It takes a while, yes.
And so, I think maybe we can kind of close it out by sharing like some things that the folks on this call, where we're not quite ready to, advocate to be testing for it, because it's got to go through a lot of process, and we all know how legislation works very slowly. But what are some things that the community can do just to, help start that process, and maybe, head towards a goal of eventually getting newborn screening for that?
Sure, definitely. And, before that, I would say something that also, resonates for everything that we have here and so far. When New York State started with this, and this has been, over the years happening, and we meet regularly, twice a year, what happened here is something that I would say has been wonderful, because we were able to put together all the people that knew something about this or was doing something for this. So now every time we have a new patient with this, as Dr. Harris was saying before, I can pick the phone if I have a question, and I can call people in Pittsburgh or Duke that usually do the bone marrow. I can call Mayo and talk to the lab there that does part of the testing. I can call Harvard and talk to Dr. Reichler that is developing gene therapy for the thing. So, and meaning that the research for the family and the patient is there. And we can put them in contact with everybody that knows about this stuff. So going back to your question, well, clearly, again, I think that everything we had been hearing is the path to get there. So basic research, translational research, advocacy. So sure, as you said, in order to eventually put something new in the list, we need to know enough. And I would agree what was said before. Probably we're never gonna know everything about all these disorders, but we might learn enough to be able to act. So if we get there, well, it might be a chance that we can just move forward into this. So if, as the other people were saying, we figure out that maybe this molecule, another molecule might work, and we can start a trial and we see some actual positive results, well, we can say, well, although we don't understand this whole thing, we find something that actually helps. And as Dr. Harris says, very important, sometimes we can target something that the disease is showing to us, maybe not everything, but if something can be improved, and the parents know this very well. So sleep problems, behavioral problems, tantrums, whatever. So these little things that look little are not little. For the parents are everything. So if a kid doesn't sleep well because he has something that affects their brains, well, this family is extremely, extremely disrupted because it's super tough. Well, if we find something that can make the kid sleep without changing anything else, the whole thing changes dramatically for them. So meaning that all these avenues that we are exploring here eventually can end up in something like might be enough to say, well, you know what, now we think that if we catch these kids early on, we can make a bigger difference than we wait until we diagnose them. And you said at the beginning that unfortunately, the average time for diagnosing these rare diseases is probably 5 years, 6 years, 7 years. And this is related with the things that, Andrew was saying. If you live in a place where you next door, you have experts and a big hospital, sure, you might get lucky. But most people don't live in these places. So they can go around forever to one office, to another office, to another office, and the doctors don't have any clue, and they don't know. So, and unless, of course, as you know, Natasha was saying, well, somebody in Spain tells you, well, listen, it's this guy over there. Well, that's why these networks actually work so well.
Sure.
Even ourselves, you know, as you know,
Mhm.
professionals have our own networks, and every time we find something strange, we post the things, and the NIH is doing a great work, you know, linking, and we can contact you guys. So I think that definitely, you know, now is a global enterprise, and we can find help anywhere in the world, and actually we're able to get there. So I think it's, you know, we are living a super exciting time, and it's huge hope for all these children, definitely.
Thank you, Dr. Iglesias. And I know we've gone over time a little, and just a few more things, you know, I can kind of speak as well as to what some of you on the call can do in this community. You know, I mentioned earlier on when I did my introduction that I oversee our Rare Action Network program, which is our grassroots advocacy network here at NORD. We work with volunteers across the country to advocate for rare diseases. Newborn screening is one of those things, but even, you know, access and, you know, equitable access, timely diagnosis. And some of the things that you all can do, you know, today is you know, join our network. You can visit rareaction.org and sign up for our network, and you'll get connected to, you know, what's going on in your state, specifically on state initiatives that NORD is working on that impact the rare disease community at large, and also federal issues as well. And some of that is overlap. And I know we talked a lot about newborn screening, and that is something that is both a federal issue and a state issue, because once it's, you know, approved on a federal level, then it's up to the states to actually initiate the testing, and that causes all different things in terms of budgets and all other aspects. So there's a lot of advocacy that needs to be done, not only in newborn screening, but in all areas for rare disease research and you know, to find cures. So, you know, I encourage you all, one thing you can do, you know, your story is the most powerful tool you have. I can't emphasize that enough. Just, you know, coming and sharing your experience in the disease space, in the rare disease space, and the challenges you've faced, and realizing that you are not necessarily alone. I mean, NORD's motto is alone you are rare, together we are strong. I like to use the analogy when I'm working with advocates that, you know, we start out in an empty stadium and we're cheering and yelling, but nobody can hear us. But if you bring together the rare disease community and the commonalities that are happening within those communities, even if they're different diagnoses, then you're filling that stadium, and then people will actually hear you. So I think this is really a great
And I also said, you know, unfortunately, you know, what happened last year and still happening has been, you know, I would say you talk about isolation of the patients. Well, it created isolation of us. So because before, you know, every time we go to a meeting, you guys are there, people from the foundation are there, everybody's there, we talk to each other, everybody can share stuff. So, but the other part is obviously we need to get out from our office and talk outside the office, because that's the only way that, you know, these things are going to be, you know, learned by eventually the people that can make decisions that help us. So absolutely, I agree, absolutely, 100%.
Great. Well, thank you again, and thank you to all of our panelists today. It was just a great panel. We've come to our time is up. I'm happy to, you know, I know the team can share my contact information if anybody would like more information on NORD, our resources, and our Rare Action Network. You can visit rarediseases.org or rareaction.org to find information on us. I just have a couple of closing items. We want to, you know, thank all of our panelists once again. You did a fantastic job. It was very educational evening tonight. And we'd like to thank the underwriters for tonight's program, Joan and Sam Ginsberg, Karen Ginsberg, and Jeremy Elkins, the CZI Chan Zuckerberg Initiative, Thank you to all of you that attended this evening. We hope that you have gained some additional knowledge and you're ready to take more action. A recording of this is going to be available on the Cat6A YouTube channel, so you can keep an eye out for that. And I do apologize we didn't get to questions this evening, but I believe the team has a way to kind of follow up with those of you that did have some questions. So thank you all. It was a pleasure to be your moderator tonight, and I hope you all have a wonderful evening.
Bye-bye, everyone.
Thank you so much.
Thank you, everyone.
Thank you. Bye.
Thank you, everyone. Have a great night.
Yes.
Thank you, Kristen. Have a good night. Thank you, everybody.
Thank you as well.